Hello, everyone. This is Qunsheng from Wuxi AppTech. First of all, I would like to thank the organizer for this opportunity to share our immuno-oncology platform established at Wuxi and how it would enable your drug discovery program. So here's my outline. So my talk will focus on a CRISPR-Cas9 mediated screening platform enabling identification of genes that control cancer cell sensitivity to the T-cell mediated elimination as a potential targets or biomarker. In the end, I will be briefly share an introductory overview of our IO drug discovery service platform, including in vitro, in vivo, and immune profiling capabilities. As all you know that despite a significant clinical activity achieved in a broad range of the tumor type with immune checkpoint inhibitors, majority of the patients do not respond to the therapy. So this drove a significant effort and investment on the combination trials. However, given the complexity of the tumor microenvironment, there is a need for systematic screens to drive discovery of drug targets that can enhance the responsiveness to checkpoint therapy. The cancer results from a genetic instability and accumulate mutations that allow cancer cell to escape intrinsic growth control and extrinsic, mostly immune constraints. So this characteristic has been well discussed in the cancer hallmark review by Hanahan and Weinberger. So systematic efforts such as Chile's project are starting to employ CRISPR-Cas9 library to screen hundreds of cancer cell lines for genes essential for cell survival. So however, essentiality of genes often changes when the cancer cell will grow under different conditions. So in the past few years, we and others have interrogated essential genes after small molecule or antibody treatment co-cultured with T cells or grown as spherules. So at Wuxi, we have established a strong CRISPR library screening platform as a service capability to work with our partners to understand the vulnerability of the cancer cell in vitro and in vivo. Our CRISPR service covers the whole spectrum, starting from customized CRISPR library design, a library synthesize, as well as QC, to the library screening in either the cell culture or syngeneic model based in vivo setting, and all the way to the sequencing and candidate gene identification with support from our dedicated bioinformatics team. So here is an internal case study in which we introduced a genome-wide CRISPR knockout library to a pomalidomide-sensitive multiple myeloma cell line. So by deep sequencing, the cell that survived from pomalidomide killing, we have identified almost all subunits of the COP9 signanosome. In addition to Cerevan, the known direct pomalidomide target, we further demonstrated that COP9 signanosome regulates the turnover of Cerevan. The data were recently published in Leukemia. So unlike pomalidomide, most drugs are not as potent in killing cancer cells. It is therefore desirable to identify cancer mutations that can sensitize such drug. This mutation can either serve as biomarkers for patient stratification or new targets to develop for combination therapy. So here is another internal case study where we introduced a genome-wide CRISPR knockout library to a pancreatic cancer cell line near PACA2 and explored genetic mutations that would cause the synthetic lethality with their cell cycle inhibitor V1 degrader. As shown on the right-hand side, we identified many cell cycle and cytokinesis regulators, validating a devastating consequence of the multiple disruption of the cell cycle or progression. So we also found 3 out of 6 heme biosynthesis enzyme from the screen, suggesting surprisingly that involvement of this metabolic pathway. So for IO target or biomarker discovery, an ideal setting would be in vivo, where the tumor microenvironment is largely intact. So, however, for the in vivo screening, we cannot use whole genome library in order to assure the sufficient guide coverage in a syngeneic tumor. Typically, we synthesize a library with less than 2,000 guides for an initial inoculation of 2 million cells to have at least 1,000-fold coverage. So in this case study, we generated a customized library targeting 313 genes that are frequently mutated in human cancers, and introduced the library to mouse MC38 cell lines. So this cell was then inoculated to immune-competent mouse host and treated with checkpoint inhibitors such as anti-mouse PD-1 antibody. So in the end, the tumor were dissected for sequencing to determine the abundance change of each individual guide RNAs. So by comparison, anti-PD-1 treated tumor with IgG treated tumor, we found guide RNAs targeting PBRM1, ARID2, KMT2D, and KDM68 were significantly deleted, suggesting the mutation in these genes would sensitize the cancer cells to the PD-1 blockade. And likewise, we also found the mutations in several other genes cause resistance to checkpoint inhibitions, but they will not be discussed here. So depletion of PBRM1 and ARID2 was already reported to sensitize cancer cell to the T-cell mediated cytotoxicity in a co-culture screen. Supporting this observation, renal cancer patients with mutations in PBRM1 showed better response to the immune checkpoint blockade therapy clinically. So we therefore hypothesized that KMT2D and KDM68 might function similarly to the PBRM1 in altering tumor vulnerability to the cytotoxic T cells. So KMT2D is a major histone monomethyltransferase and regulates enhancer activities and cell type specific gene expression. So KMT2D is frequently mutated in many cancer cells, and Kabuki syndrome, a rare genetic disease. So we validate the screen hits. To validate the screen hits, we perform in vivo competition assays by mixing the GFP expressing KMT2D knockout cells with cherry-expressing control cells and implanted them to immune-competent mice. So after treated with anti-PD-1 or PD-L1 antibody, tumor was dissected and the fluorescent cell were analyzed by the flow cytometry, which is showing on the left-hand panel. So we found by surprise that KMT2D knockout cells gain a growth advantage over control cell, especially in the in vivo setting. And this growth advantage could be offset by immune therapy. We're currently still testing an triggering hypothesis that loss of KMT2D in cancer cell enable PD-1 dependent immune escape. So in addition to the CRISPR-mediated target identification and validation capability, we also build a comprehensive drug discovery service platform enabling downstream drug testing. So this includes in vitro immune function assays, in vivo pharmacology using the syngeneic model and the humanized model, as well as immune profiling capability of the FACS, immunohistochemistry, multiplex immunofluorescence, nanostream, and next generation sequencing technologies. So for the interest of time, I will quickly go through them as a snapshot for the rest of my talk. So the table shown on the left is a list of established in vitro cell-based assay covering major immune functions. So most of these assays are using the human PBMCs. So in addition, we also use mouse or non-human primate PBMCs in parallel for bridging a different species. And on the right-hand panel side, a panel of markers of the mouse tumor infiltrating leukocyte, TILs, were established and validated using a standard flow cytometry. So besides the TILs, the other markers such as checkpoint co-stimulating markers as well as cytokines are also available. So we have established a panel of 61 syngeneic tumor models and accumulated quite experience in IO-related pharmacology services. So including the tumor microenvironment assessment as a PD readout. So as shown in panel A, out of the 61 syngeneic models, we extensively profiled about 220 models in terms of their sensitivities to checkpoint inhibitors and baseline of RA expression and the TILs. The panel B shows a few examples of PD-1 or PD-L1 in combination with other checkpoint modulators or the radiation therapy. So the panel C and D are tumor profiling by flow and immunofluorescence staining following the combination study. So as mentioned earlier, we took quite efforts to build immune profiling capabilities with a goal to work with our clients for understanding mechanism of action and the discovery of potential biomarkers. So besides the flow immunohistochemistry and the multiplex IF, we have established nanostream single cell RNA sequencing of the TIL as well as TCR repertoire sequencing capabilities as shown on this slide. And due to species cross-activity issues, there is need for humanizing mice for assessing immune modulation of the large molecule. As you know, there are two commonly used approaches of humanization: human PBMC adoptive transfer, showing on the left, and CRISPR-mediated genetic humanization of specific target gene shown on the right-hand side. So both systems are readily available at WuXi. We have established about 56 xenografts on the top of human PBMC adoptive transfer mice. So using the genetic knock-in approach, a number of humanized mice were generated covering single, double, or triple humanized gene as shown on this table. So for example, here is our internal efforts that validated human PBMC adoptive transfer xenograft model by conducting an anti-tumor efficacy study with a bispecific antibody Blincyto. We also have experience in emerging IO modalities, including CAR-T, oncolytic virus, and vaccine. So for the interest of time, I only included an example of screening cancer vaccine using an ex vivo ELISpot platform, and his workflow shows on the left hand. So following ELISpot screening, any promising candidate peptide, for example, can further be characterized for his mode of action by depletion of CD4 or CD8 cells using the flow cytometry. So for therapeutic vaccine, anti-tumor efficacy is also part of testing cascade. So now I'm switching gear to clinical biomarker capabilities at the WuXi. So our pathology and flow lab are certified by College of American Pathologists and GCP compliance for enabling clinical trials. So the lab equipped with board certified pathologists, state-of-art instruments, and Hello digital pathology. So providing one-stop shop service with rapid turnaround. So in addition to standard immunohistochemistry and IF, we also perform molecular pathologies such as FISH, DISH, and RNAscope. So this is a snapshot on IO-related biomarker that were established and certified, showing on the left. I also shows an example of detecting c-Met alteration, including exon 14 skipping mutation by using the RNAscope. So we have over 100 clinical validated biomarker that available, which are not shown here. So before conclude this talk, so I would like to quickly highlight our accreditation and other capabilities that I didn't have a chance to touch upon. So WuXi Oncology and the Immunology Unit has over 500 dedicated research scientists. So we are operating in China with capacity of about 200,000 square foot lab facility, including the ALAC certified SPF vivarium, equipped with 30,000 IVC cages for the rodent study. So currently, there are around 350 active partners around the globe. So this is my last slide, highlighting our service covers target identification validation all the way to the clinical biomarker testing. So I showed you our IO-related capabilities, but we also have a comprehensive capability to enable target oncology, autoimmune disease, and rare genetic disease drug discovery. So for the targeted oncology, a large panel of the CDX, PDX, and orthotopic models, alongside with 50+ drug-resistant tumor model, are readily available at WuXi. So for autoimmune disease drug discovery, there are about 26 related disease models. We also provide genetic engineering animal model plus pharmacology services to support rare genetic disease drug discovery, including the AAV packaging service for gene therapies. So besides standard small molecule and biologics, we have rich experience in emerging modalities such as PROTAC, ADC, CAR-T, oncolytic virus, etc. I stop here. Thanks for your time and listening. I would be happy to take any question.